Ever pondered the intricacies of DNA sequencing? How do scientists unravel the secrets locked within the DNA, the blueprint of life? Today, we dive into the fascinating world of Sanger DNA sequencing. Named after its developer, Frederick Sanger, a British biochemist and two-time Nobel laureate, this method revolutionized the field of genetics when it was introduced in the late 70s. DNA, as we know it, is composed of 4 nucleotides: adenine, guanine, cytosine, and thymine. These building blocks link together to construct the iconic double helix. In this structure, adenine forms a pair with thymine, while guanine shares its bond with cytosine. This specific pairing method is integral, lending stability to the helix. More importantly, the particular sequence of these nucleotides forms the basis of our genetic identity. It's this precise arrangement that creates our one-of-a-kind genetic code. So how does Sanger sequencing decipher this code? This method, also known as chain termination method, starts with a single strand of DNA. This strand acts as a template for synthesis of a new DNA strand. First, the DNA is heated to separate the 2 strands. Then a primer, a short strand of DNA, is added. The primer binds to the start of the section of DNA we want to sequence. Next, DNA polymerase, an enzyme, is added. This enzyme starts adding nucleotides to the primer, creating a new DNA strand. Here's where the magic happens. Along with the regular nucleotides, modified nucleotides called dideoxynucleotides are also added. These dideoxynucleotides are special. They are similar to regular nucleotides, but lack a specific group called the 3-hydroxyl group. This means that once a dideoxynucleotide is added, no more nucleotides can be added. The DNA strand is terminated. Each type of dideoxynucleotide is labeled with a different color. For instance, the dideoxynucleotide for adenine might be green, while that for thymine might be red. As the DNA polymerase adds nucleotides, each time a dideoxynucleotide is added, a differently colored fragment is created. This process creates many fragments of different lengths. Then, these fragments are separated by size using a process called electrophoresis. The smallest fragments move the fastest, and the largest ones move the slowest. As they pass a detector, the color of each fragment is recorded. This gives us the sequence of nucleotides in the DNA strand. In summary, Sanger sequencing involves creating fragments of DNA of different lengths using modified nucleotides, separating these fragments by size, and then reading off the sequence. This method has been instrumental in many scientific breakthroughs, including the Human Genome Project. It's a testament to the power of scientific curiosity and ingenuity, and of course, to the marvel that is DNA. So the next time you marvel at the complexity of life, remember the simple yet powerful technique of Sanger DNA sequencing that helps unravel its secrets, one nucleotide at a time.